Cholesterol Synthesis by Jaime Coverti. Cholesterol is one of the most publicized lipids, well known because of the strong correlation between high levels of cholesterol in the blood and frequent human cardiovascular diseases. Less advertised is cholesterol's crucial role as a component of cellular membranes and as a precursor of steroid hormones and bile acids. The structure of this 27-carbon compound found here suggests a complex biosynthetic pathway, but all of its carbon atoms are provided by a single precursor, known as acetate. The isoprene units that are the essential intermediates in the pathway from acetate to cholesterol are also precursors to many other lipids. Cholesterol, as we all know, is made from acetyl-CoA, but the assembly plan of cholesterol is quite different from that of long-chain fatty acids. The synthesis takes place in 4 stages, shown here. The condensation of 3 acetate units to form a 6-carbon intermediate, mevalonate. The conversion of mevalonate to activated isoprene units. The polymerization of 6 5-carbon isoprene units to form the 30-carbon linear squalene. And the cyclization of squalene to form the 4 rings of the steroid nucleus, with the many oxidations and the removal of migration of methyl groups to produce cholesterol. The first stage in cholesterol biosynthesis is the synthesis of mevalonate from acetate. 2 molecules of acetyl-CoA condense to form aceto-acetyl-CoA, which condenses later with a 3rd molecule of acetyl-CoA to produce beta-hydroxy-beta-methylglutaryl-CoA, also known as HMG-CoA. The first 2 reactions are then catalyzed by acetyl-CoA acetyltransferase and HMG-CoA synthase. HMG-CoA synthase then catalyzes HMG-CoA synthesis in yielding the ketone body formation. We later then reach the committed step, which is the reduction of HMG-CoA to mevalonate. 2 molecules of NADPH each donate 2 electrons. The second stage in cholesterol biosynthesis is the conversion of mevalonate to 2 activated isoprenes. To begin this, we have 3 phosphate groups that are transferred from 3 ATP molecules to mevalonate. Both this phosphate and the nearby carboxyl group leave, producing a double bond in the 5-carbon product known as isopentyl pyrophosphate. This is the first of the 2 activated isoprenes central to the cholesterol formation. Isomerization later then yields the second activated isoprene, dimethylallyl pyrophosphate. The third stage in cholesterol biosynthesis is the condensation of 6 activated isoprene units to form squalene. Disulpentyl pyrophosphate and the dimethylallyl pyrophosphate perform a head-to-tail condensation, where one of the pyrophosphate groups is disabled and a 10-carbon chain, geranyl pyrophosphate, is later formed. Geranyl pyrophosphate undergoes another head-to-tail condensation with isopentyl pyrophosphate. This then yields the 15-carbon intermediate, farnesyl pyrophosphate. To conclude, 2 molecules of farnesyl pyrophosphate join head-to-head, with elimination of both pyrophosphate groups to form squalene. The final stage of cholesterol biosynthesis is the conversion of squalene to the 4-ring steroid nucleus. Squalene adds 1 oxygen atom from O2 to the end of the squalene chain. This then forms an epoxide. This enzyme is another mixed-function oxidase. NADPH reduces the other oxygen atom of O2 to H2O. Squalene 2,3-X epoxide is positioned superbly to convert the linear squalene epoxide to a cyclical structure. In most animal cells, this cyclicalization results in the formation of linosterol, which contains the 4 rings characteristic of the steroid nucleus. Linosterol is finally converted to cholesterol in a series of about 20 reactions, which happen to include the migration of some methyl groups and the removal of others. Finally, I would like to point out a picture found in our textbooks, depicting that cholesterol has several fates. Most of the small fraction of the cholesterol that is made is incorporated into the membranes of hepatocytes, but most of it is exported in one of the 3 forms, as bile acids, biliary cholesterol, or cholesterol esters. Okay, now quiz time. Yes, I know. Get your eye clickers ready. You have 30 seconds. In stage 1 synthesis of mevalonate from acetate, 2 molecules of acetyl-CoA condense to form aceto-acetyl-CoA, which condenses with a 3rd molecule of acetyl-CoA to yield the 6-carbon compound HMG-CoA. These first two reactions are catalyzed by acetyl-CoA and acetyl-CoA acetyltransferase and HMG-CoA synthase. The third reaction is the committed step, the reduction of HMG-CoA to mevalonate, for which two molecules of NADPH each donate 2 electrons. HMG-CoA reductase, an integral membrane protein of the smooth endoplasmic reticulum, is the major point of regulation on the pathway to cholesterol, as we shall see. Cholesterol has several fates. Most of the cholesterol synthesis in vertebrates takes place in the liver. A small fraction of the cholesterol made there is incorporated into the membrane of hepatocytes. Most of it is exported in one of three forms: bile acids, or bile salts, biliary cholesterol, or cholesterol esters. Small quantities of oxysterols, such as 25-hydroxycholesterol, are formed in the liver and act as regulators of cholesterol synthesis.